Hi everyone, this is Sivanagamani. Now I am going to explain DNA ligase. First of all, we will know about what is ligase. A ligase is an enzyme that can catalyze the joining of two molecules by forming a new chemical bond. This is typically via hydrolysis of a small pendant chemical group on one of the molecules. Now, DNA ligase. DNA ligase is a type of enzyme that facilitates the joining of DNA strands together by catalyzing the formation of a phosphodiester bond. It plays a role in repairing single strand breaks in double-X DNA in living organisms, but some forms may specifically repair double strand breaks. Single strand breaks are repaired by DNA ligase using the complementary strand of the double helix as a template, with DNA ligase creating the final phosphodiester bond to fully repair the DNA. DNA ligase is used in both DNA repair and DNA replication. In addition, DNA ligase has extensive use in molecular biology laboratories for recombinant DNA experiments. Purified DNA ligase is used in gene cloning to join DNA molecules together to form recombinant DNA. Coming to mechanism of DNA ligase. The mechanism of DNA ligase is to form two covalent phosphodiester bonds between 3' hydroxyl ends of one nucleotide, which is acceptor, with the 5' phosphate end of another, which is donor. DNA ligase reaction. Here ATP is required for the ligase reaction, which proceeds in 3 steps. Adenylation of a residue in the active center of the enzyme. Pyrophosphate is released. Transfer of the AMP to the 5' phosphate, so called donor. Formation of pyrophosphate bond. Formation of a phosphodiester bond between the 5' phosphate of the donor and the 3' hydroxyl of the acceptor. Bacteriophage T4 DNA ligase. T4 DNA ligase is an enzyme that is encoded by the bacteriophage known as T4. In a reaction where the DNA molecules are being joined together at the 3' hydroxy and 5' phosphate termini, the ligase is used as a catalyst. Given that there are no missing nucleotides in the repair reaction, the ligase can also catalyze the covalent joining of two segments to one unit erupted strand in a DNA duplex. Coming to applications: ligation of cohesive ends, ligation of blunt-ended termini, ligation of synthetic linkers or adapter. E. coli DNA ligase. It is derived from E. coli cell and required NAD+ as cofactor. It is a monomeric enzyme of molecular weight of 74 Kelvin Dalton, which catalyzes the formation of the phosphodiester bond in duplex DNA containing cohesive ends. This enzyme has narrower substrate specificity, making it a useful tool in specific application. Coming to applications of E. coli ligase. Ligation of cohesive ends. E. coli DNA ligase is used to catalyze sticky end ligation. Cloning of full-length cDNA. E. coli DNA ligase has been employed in a procedure for high-efficiency cloning of full-length cDNA. Tag DNA ligase. The gene encoding thermostable ligases have been identified from several thermophilic bacteria. Several of these ligases have been cloned and expressed to high levels in E. coli. It is uses in the detection of mutation, as thermostable DNA ligase retain their activities after exposure to higher temperature for multiple rounds. It is uses in DNA amplification reaction to detect mutation in mammalian DNA. T4 DNA ligase. T4 DNA ligase is the only phase RNA ligase that has been extensively characterized and used in genetic engineering. This enzyme catalyzed the phosphodiester bond formation of RNA molecule with hydrolysis of ATP to PPI. It is monomeric enzyme with 373 deduced amino acid residues is a product of the T4 gene 63. Coming to applications, 5' tailing of DNA with the RNA, radio labeling of 3' end of RNA and DNA, production of elongated molecules, modification of internal nucleotide, stimulation of T4 DNA ligase activity. This is all about DNA ligase. Thank you.